Hello and good evening, viewers. Today we are going to talk about introduction to enzymes. So to discuss this topic, we have with us in our studio Dr. Amit Bhattacharya, and he is assistant professor at Ramjas College, University of Delhi. So without wasting any time, I would like to welcome our guest, Dr. Amit Bhattacharya. Sir, welcome to EduSet Live lecture.
Thank you very much, ma'am. So in this part of the lecture, I will be talking about what exactly the enzymes are, how exactly the enzymes functions, and what are the roles of the various active sites, how the inhibitors helps in the blocking or the feedback of these enzymes. Subsequently, I will talk about the enzyme kinetics and the various classification of the enzymes. Before going into the enzymes part, we should know what exactly the enzymes are. So enzymes are basically, they are made up of proteins. So the enzymes have got few of the characteristic feature which includes the catalytic power and specificity. Subsequently, the catalysis within the enzymes takes place at a specified location which are called as the active sites. So nearly all the enzymes are proteins. There are some exceptions like the lysozymes and ribozymes. Now, the enzymes are made up of the protein molecules. So all of us know that the proteins are basically the chain of the amino acids which are joined together to one another with the help of the peptide bonds, and they interact in a three-dimensional structure to formulate or form the enzyme structure. Now, how exactly these protein chains or the polypeptide chains assembles into each other? So basically the protein chains or the amino acid chains are called as the primary structure of any amino acid. Now these primary amino acid structure or the chain subsequently build up to the secondary structure, which are basically the helix structure or the beta pleated sheath. Subsequently, the secondary structure folds further into the tertiary structure, and the tertiary structure subsequently folds into the quaternary structure, which are the assembled subunits of the enzyme. Now, if we see the central dogma, how exactly these proteins are formed within the cells. Now, we know that the DNA which is present within the nucleus, it is transcribed to form the RNA. Subsequently, the RNA is processed, and this processed RNA moves out from the nucleus into the cytoplasm. Within the cytoplasm, now the next phase of the reaction start taking place. Now, if we see in the cytoplasm, the mRNA have moved out and it goes and gets attached to the ribozyme. Within the ribozymes, there are specific locations in which the codons brings up the specific polypeptide or the amino acid chains. Now subsequently, the amino acids are attached to one another and they form the polypeptide chain, decoding the mRNA which has been attached to these ribosomes. So this is how exactly these protein structures are formed within the body cells. Now, the enzymes are classified mainly into major categories. The first is the catalysis for the rate enhancement. So all the enzymes do this to a certain extent. Then there is a specificity for the substrate. So the enzymes are very specific for their substrate. Third is regulation of reactions or the pathways. So the enzymes modulates or controls the various reactions pathway which are very important for the body functioning and the metabolism. Then the enzymes have got a very important part which is called as the cofactors or the co-substrates, which we subsequently will discuss in this enzymes presentations. Now in this enzymes part, we will discuss about what exactly the enzymes kinetics is, and then we will go into what happens in these enzymes. Now, we have to discuss what exactly these enzymes does. So, let's see some of the examples how these enzymes function. So, over here I have shown an example why do fireflies light up. So, this is a reaction which is taking place with the help of the enzyme. Now, in this, the enzyme which is functioning is called as the luciferase. So what happens is, in the fireflies, the luciferin substrate with the help of the luciferase enzymes breaks down or gets converted into the product which is called as the oxyluciferin. In this process, there is an emission of the light which is emitted by these fireflies. So we can see in this image the firefly produces a chemical reaction inside their body that allows them to light up. Now we take up another example in the case of the jellyfish. Now if we see, the jellyfishes also emit a certain kind of light. So what happens is, this takes place with the help of another enzyme which is present within the jellyfishes, which is called as the aquaporin. Now this aquaporin enzymes is responsible for the emission of the light. Now for this breakthrough work in the year 2008, the Nobel Prize were given to three of the scientists, Shishimura, Chalfrey and Cezanne. Now they were instrumental in the discovery and the development of the green fluorescence protein, or the GFP protein. So what they found is, the jellyfishes generally glow with the help of certain enzymatic reaction. Now they first isolated one of the bioluminescent protein that gave up the blue color, but subsequently they found that the jellyfishes were emitting green color light. So they got interested and they found that there is a second jellyfish protein. What this protein does is, it absorbs the blue color light and transmits or re-emits the green light. So they named it as the green fluorescence protein, and for the discovery and the isolation of this green fluorescence protein, they were awarded the Nobel Prize. Now how exactly these enzymes functions within our body? So in our body there are different metabolic pathways which function. Now over here I have shown a picture of how exactly the carbohydrate, proteins and the lipids or the fatty acids gets degraded. Now the carbohydrates, we know that, are made up of long chains of the glucose or the various saccharides. Now these saccharides, with the help of the various enzymes within our body, they get broken down into the small monosaccharides, which are basically the glucose molecule. Subsequently, the glucose molecule goes into the various metabolic pathways and they start functioning or provide the energy for the cells. Now in the case of the amino acids, we know that the amino acids comes from the polypeptide chain or the protein molecule. Now this protein molecule is made up of a number of amino acids. So over here a number of amino acids has been shown which are linked to one another with the help of the polypeptide chain. Now with the help of the various enzymes which is present within our body, these amino acids are degraded or chopped off into the subsequent amino acids, individual amino acids. Now in the case of the fatty acids or the lipids, these fatty acids and the glycerols are separated with the help of the various enzymes such as the lipases. So these lipases, what they does is, they breaks down the fatty acids or release these fatty acids from the glycerol backbone. Now how exactly we digest the food? So we digest all the foods with the help of the enzymatic activity. Now over here I have shown a diagrammatic representation of the gastrointestinal tract or the digestive system. So at the various places or the various organs within this gastrointestinal tract, the various enzymes are produced, and these enzymes helps in the digestion or breaking down of the food which transfers from the mouth into the large intestine. Now if we narrow down or zoom in into what exactly happens in the various structures or the various organs of these gastrointestinal tract or the digestive system. So as soon as we ingest the food from the mouth, the salivary glands transfers the salivary amylase which has been produced within the salivary gland. Now this salivary amylase forms the first line of breaking down of these food molecules. Subsequently they go into the stomach, then subsequently from the small intestine to the intestine, where the various forms of the enzymes are produced which helps in the digestion and breaking down of these food sources. Now if we see the tabular representation over here, this tabular representation shows how exactly the carbohydrate, proteins, nucleic acid and the fatty acids are digested at the various parts of our body. So in the case of the carbohydrate digestion, if we see in the oral cavity, pharynx and the esophagus area, with the help of the salivary amylase, the polysaccharide is broken down into the smaller polysaccharides or the maltose. Now within the small intestine there are pancreatic amylase which are poured in. So they subsequently breaks down these polysaccharide into the disaccharides such as the maltose and the other disaccharides, and within the small intestine epithelium areas, these disaccharides are further broken down with the help of the disaccharidase enzymes to break down into the monosaccharide. While in the case of the protein digestion, if we see, there are different enzymes which are produced, such as from the stomach there is a presence of an enzyme which is called as the pepsin. So the protein is broken down into the small polypeptide chain with the help of the pepsin. Subsequently, these small polypeptides are further broken down in the small intestine with the help of the enzymes which is called as the trypsin, chymotrypsin, and subsequently these smaller polypeptides are further broken down into individual amino acids with the help of the aminopeptidase, carboxypeptidase, and these are further broken down with the help of the dipeptidase in the epithelium of the small intestine also. In the case of the nucleic acid digestion, what happens is within the small intestine there is the presence of an enzyme which is called as the nuclease. So these enzymes helps the breaking down of the DNA or the RNA into the nucleotides. Subsequently, the nucleotidase enzymes breaks down these nucleosides into the nitrogenous bases, sugars and the phosphates, which forms the backbone of the nucleic acid chain. Now, in the case of the fat digestion, what happens is the fat globules are first broken down into the fat droplets, which are the emulsified formulation. These takes place with the help of the bile salts. Subsequently, the lipase enzyme breaks down the glycerol and the fatty acids or the glycerides. So this is how exactly the carbohydrates, proteins, nucleic acid and the fatty acid digestion takes place. Now, let's come into the enzyme kinetics, how exactly these enzyme functions and what exactly how these enzymes are controlled by the various factors. Now, before going into the enzyme kinetics, we should know what exactly happens in the enzyme substrate interaction. So what happens is when an enzyme interacts with the substrate, there is a formation of a transient state molecule which is called as the enzyme substrate complex. Now this enzyme substrate complex subsequently goes into the breaking down of the substrate into the product form and the product is released. So the total reaction is said to be E plus S gives ES, which further gives the catalytic step, which is the E plus P. So the product is released from the enzyme part. So we can see the enzyme function is divided into two major steps. The first is the substrate binding step, the second one is the catalytic step. Now, how exactly the enzyme function? So all the enzymes have got a active site in which the reactions takes place. So the enzyme active site, to the enzyme active site, the substrate comes and binds to this one. The reactions start taking place and subsequently the reaction goes off and the products are released from these active sites. So the active sites of an enzymes are a particular and specific location where the reactions start taking place. Now, if we see the molecular imaging of how exactly the active sites looks like, so over here I have shown an active site to which the substrate has got bound. Now, these active sites are basically the restricted regions or the areas of the enzyme molecule which binds to the substrate. So these are basically the pockets or the grooves within the enzymes on the protein surface, and they are formed with only a few of the amino acids from the enzyme polypeptide chain. Now, if we see over here, the enzyme polypeptide chain are shown, and within this polypeptide chain, certain amino acids interacts with each other to form the active site in which all the reactions start taking place. So if we linear this polypeptide chain, we can find that all these amino acids are located at a distant place from one another. So by the three-dimensional structure of the enzymes or the polypeptide chains of the enzymes, all these amino acids which are present at the different location within this polypeptide chain comes in a close proximity and they interact to one another to form the enzyme active site. Now, over here, if we see the image over here, in this image I have shown the enzyme substrate complex and how exactly the enzyme substrate complex looks like in a three-dimensional structure. Now, the enzyme functioning was given in two basic models. The first model which came up was called as the lock and key model. This was discovered or given by the scientist Emil Fischer in the year 1894. So what he formulated is the enzyme functions by a property which is called as the lock and key. So the enzyme functions as a lock and the key is the substrate. So the enzyme active site and the substrate has got a complementary structure to one another which fits to one another. So if we see the diagrammatic representation over here, the enzyme is the lock while the key is the substrate. So the key comes and binds to this enzymes and the lock key complex is formed. So the substrate is complementary to the structure of the active site which is present within the enzyme, and the substrate comes and binds to this enzyme structure to form the enzyme substrate complex. But subsequently, this the lock and key model was found to be rigid, so it was not recommended, or the scientists didn't follow this model system. Subsequently, another model system came into the picture which was called as the induced fit model system, which was given by Dr. Daniel Koshland in the year 1963. In this model system, what is recommended is the enzyme active site and the substrate don't have a complementary structure. What happens is, as soon as the substrate comes in a near proximity to the enzyme's active site, the enzyme active site dimensional structure changes. So there is a conformational changes which takes place in the active site, and subsequently this active site takes a position which is complementary to the substrate structure. So over here he proposed that the enzyme active site is induced to fit with the substrate which is coming and getting attached to that particular active site. So if we see the structure or the image over here, the enzyme active site is shown over here. So in this image, if we see in this image, we can see the substrate and the enzyme active site which is not in a complementary structure with the enzyme substrate. As soon as the substrate comes near this enzyme active site, there is conformational changes which takes place in the enzyme substrate complex structure, and the enzyme active site gets into a complementary structure which fits into the substrate molecule. Now, the enzyme kinetics basically follow some of the basic rules. So the enzyme kinetics, what happens is, as soon as the enzymes get attached to certain reactions, it lowers the activation energy. So if we see in the graphical representation, the red color line shows a uncatalyzed reaction where there is no enzyme which is involved. So we can see the red color line, there is a reactant which goes into the formation of the enzyme substrate complex, and subsequently the catalytic reactions start taking place and the products are released. But a blue color graph which is shown over here is a reaction which is taking place with the help of the enzymes or the catalyst. So we can easily see over here there is a lowering of the activation energy which is taking place by the addition of the enzymes, and this is what is important in the catalysis of the reaction. Now, the question which comes to our mind is how exactly these enzymes lowers the activation energy. So basically the enzymes holds the substrate in such a way so that it can distort the substrate bondings closer to their form in the transition state. So this reduces the amount of energy which is needed to complete the transition reaction. Subsequently, the enzymes create a charge distribution opposite to that of the transition state. This lowers the energy of the transition state and decreases the activation energy. Now, there are certain other probable reasons which have been also formulated. Another of the reasons how exactly the enzymes lowers the activation energy is the enzymes reduces the reaction entropy or the delta S by bringing the substrate together in the correct orientation to react. So as soon as the substrate comes in a correct orientation to react, there is lesser amount of energy which is required for changing the orientation of the substrate. Subsequently, the enzyme provide a complete differential different chemical pathway for the reaction, it may form new bond in the enzyme substrate complex that would be difficult to form without the presence of the enzymes. So these are the four major reasons why the enzymes lowers the activation energy. Now, if we see one of the example of the rate enhancement with the help of the enzymes, we see the example of the carbonic anhydrase. So over here, if we see the carbonic anhydrase enzymes is one of the most important enzyme which is present in our body. It helps in the formation of the carbonic acid by combining the carbon dioxide and the water molecule. So in an uncatalyzed rate of reaction where there is no enzymes which is involved, there is only 1.3 into 10 to the power minus 1 molecule per second of enzymes molecules or the products which is produced. While in the case of the catalytic reaction where the enzymes are involved, about 10 to the power 6 molecule per second products are formed in this one, or the carbonic acid molecules are formed. So we can see by the presence of these enzymes, the rate enhancement has increased to 7.7 into 10 to power 6 times by just presence of the enzymes, which is the carbonic anhydrase. So enzymes play a very important role in the metabolic or the various functioning reactions within our body system. Now let's come into the enzyme kinetics. So the enzyme kinetics, if we plot a graph of the enzyme kinetics between the initial rate reaction and the substrate concentration, we can see a parabolic graph like this one. So at the initial stage, there are lots of enzymes, but there is a scarcity of the substrates which is present over there. Subsequently, as the reaction paces up, what happens is all the enzymes are attached with the substrate, or we can see that the enzymes are saturated. So the enzymes are saturated, so there are far more substrate that can deal with all the enzyme. Subsequently, the reaction reaches a maximum velocity where the substrate concentration increases or overloads the concentration of the enzymes, and this is the capacity which the enzymes can function maximum. So this is called as the maximum velocity or Vmax. Now, if we plot this particular reaction into a graphical representation, we can see over here a graphical representation where it is shown that E plus S gives rise to ES, which is the transient molecule or the enzyme substrate complex, which subsequently goes into the formation of the enzyme product complex. So over here the reaction velocity and the substrate concentration is shown over here, and we just discussed as soon as the reaction start taking place, it reaches a maximum velocity which is called as a maximum velocity or Vmax. Now there are different factors which have got an effect on the enzyme kinetics. Few of them are temperature, pH and the substrate concentration. It has been seen that all the enzymes has got an optimum temperature, or at an optimum temperature the enzyme functions maximum. So over here we can see an enzyme which is functioning more or less in a better way at a temperature which is between 30 to 40 degrees Celsius. So this temperature is the optimum temperature for the functioning of the enzyme. As soon as the temperature increases, what happens is, we know that most of the enzymes are made up of proteins, so these proteins get denatured and the enzymes stop functioning. If you see the second example with the help of the pH, all the enzymes have got a characteristic pH at which they function. Because these enzymes are made up of amino acids, so the pH is a very important factor for the enzyme kinetics. And over here the last example is the substrate concentration. So all the enzymes have got a particular substrate concentration after which it cannot function anymore. Now if we see the enzymes, the functional part of the enzymes are basically called as the hollow enzyme. So the hollow enzymes is the active enzyme. This hollow enzyme is basically made up of two parts. One is called as the apoenzyme, which is the protein part, and the non-protein part, which is called as the cofactors or the co-substrate. So the co-substrates or the cofactors are very important part of the enzymes, and they have to be attached with the enzyme to make it functional. Now let's see some of the cofactors or coenzymes and the metal complexes. So over here we discussed that the apoenzymes are basically the protein part while they are attached to a coenzymes or the cofactors, which are the non-protein portion, and they combine to form the holoenzymes to which the coenzymes or the cofactors are attached, and this holoenzyme is the active enzyme to which the substrate can bind, and subsequently the catalytic reaction can start taking place. Now, if we see some of the example over here, the coenzymes, we can see there are different enzymes like thymine pyrophosphate, which is a coenzyme for the pyruvate dehydrogenase. Then there is biotin, which is a enzyme for the pyruvate carboxylates. Then there are different metals such as the zinc, magnesium, nickel, potassiums, which helps in the enzyme functionings, like the carbonic anhydrase has got the metal cofactor, which is the zinc. Then there is urease, hexokinase, which are the very important metabolic enzymes which requires the magnesiums and the nickel for their function. So we can see the enzymes for their proper functioning require certain cofactors or the coenzymes or the metals for its functioning part. Now, the enzymes have been classified into various categories based on what exactly the reactions they do. Now, the 6 major enzyme classification categories are the oxidoreductase, where the first one are the enzymes which basically does the oxidation and the reduction processes. So, the example over here is the lactate dehydrogenase. The second major class of the enzymes is the transferases, which transfers the groups from one molecule to the other molecule. The example over here is the nucleoside monophosphate kinase. The third example is hydrolases, which hydrolyzes the reactions, and the example is chymotrypsin. The fourth one are called as the lyases, which adds or removes the group to form the double bonds. Over here, the example is fumarase. The fifth one is the isomerase, which does the intramolecular group transfer or the isomerization reaction. The example is the glycolytic reaction enzyme, which is triose phosphate isomerase. And the sixth one is the ligases, which ligates to substrate at the expense of ATP. The example is aminoacyl tRNA synthase. Now, over here, the first example I have shown is the oxidoreductase over here, which brings about the oxidation and the reaction. So, the lactate dehydrogenase is one of the enzymes which falls under this category. Over here, the lactate is converted to the pyruvate by the oxidoreduction reaction, where the NAD is reduced to NADH plus H plus. The second major class of the enzymes is the transferases, where the alanine aminotransferase transfers the alpha amino group between the alanine and the aspartate. So we can see the alpha amino group which is transferred from the alanine to the aspartate with the help of the alanine aminotransferase. The third class of the enzymes are called as the hydrolases. Over here, there is a use of the water molecule. The fourth class of the enzymes are called as the lyases. Over here, there is a use of the water which are the fumarases. So the fumarate, with the help of the water molecule, gets converted to malate. The fifth class of the major enzymes classification is the isomerases. So they bring about the intramolecular group transfers. Over here, if we see the dihydroxyacetone phosphate gets converted to glyceraldehyde 3-phosphate with the help of the triose phosphate isomerase. And the last or the sixth enzyme classification category is the ligases, which brings about the ligation of two molecules, and the example over here is the tRNA synthetases. Now, the enzyme kinetics was mainly discovered, or it was given by two scientists, Michaelis and Menten. So they discovered or hypothesized one of the enzyme reaction which is commonly known as the Michaelis-Menten reaction. So this Michaelis-Menten reaction is basically a rate catalytic reaction in which defines the rate of catalysis, which is the V naught, as the substrate or the number of moles of product formed per second, which varies with the substrate concentration. So this Michaelis-Menten enzyme kinetic reaction, what it gives us the idea is, it gives us the idea of rate catalysis, which rises linearly as the substrate concentration increases and then begins to level off and approach a maximum at the higher substrate concentration. So if we see the graphical representation over here, this is called as the Michaelis-Menten plot. We can see that initially the rate of reaction increases with the reaction velocity or the V naught. Subsequently, it increases and reaches a plateau where the reaction rate is maximum, and this reaction rate maximum is called as the Vmax. The Michaelis-Menten equation is called as the V naught is equal to Vmax into S, which is the substrate concentration, divided by the Km, which is the Michaelis-Menten constant, plus S. So over here, with the help of these graphical representation, they can deduce that Michaelis-Menten reaction is the substrate concentration when the reaction is half maximum, or the Km is half Vmax. Now, if we discuss the Michaelis-Menten constant further, so Michaelis-Menten constant, as I said, is the substrate concentration when the initial velocity or the V naught is half maximum. So the lower value of Km shows that there is a tighter substrate enzyme binding which is taking place. So Km is a measure of affinity of the enzyme for the substrate. So we will see some of the examples how exactly the Km values differ. Now over here I have shown some of the examples of Km values of some of these enzymes. So if we see, the chymotrypsin has got a very high Km, which shows that there is a lesser amount of substrate enzyme binding or the affinity, while there are certain enzymes such as the arginine tRNA synthetase. Then there is different enzymes like the penicillase enzymes which have got a low Km value, which shows that, like the pyruvate carboxylase, which have got a low Km value, which shows that they have got a higher affinity to that particular substrate for the enzyme. So the Km value is giving us an indication about the affinity of the enzyme for the substrate. Now the Michaelis-Menten reaction further gave into another plot which was known as the LineweaverBurk plot. Now in the Michaelis-Menten plot, or through the Michaelis-Menten equation, the scientists were unable to find the exact value of the Vmax or the maximum velocity, because at the maximum velocity, we just saw the graph, the velocity reaches the plateau or the asymptote region. So the scientists were unable to find what exactly the maximum velocity were. So what they did is they reciprocated both the sides of the Michaelis-Menten equation, and this was subsequently known as the LineweaverBurk plot or the double reciprocal plot. So over here, the initial reaction which was in the Michaelis-Menten equation became 1 by V naught is equal to Km divided by Vmax into S plus 1 by Vmax. So this equation is something like a mathematical equation which shows is y is equal to mx plus c, where m is the slope and the c is the x-intercept. So if we plot a graphical representation from this particular equation, we get a graph or the plot like this, where the intercept which is shown, which is minus 1 by Km, while the slope is Km by Vmax, which is crossing the 1 by V naught intercept over there, which is 1 by Vmax. So with the help of this LineweaverBurk plot, the scientists were able to find out the exact value of the Vmax, and this became one of the breakthrough or the additional equation along with the Michaelis-Menten equation, and this was known as the double reciprocal plot or the LineweaverBurk plot. Now with the enzymes, there are a number of definitions which are involved. One of the most important terms which are involved with the enzymes is the turnover number or the kcat value. So in an enzyme, the catalytic reaction or the overall reaction of the product formation is equal to V is equal to K2 by ES. If we see the equation over here, the K1 and K minus 1 are the rate constant, where the enzyme plus the substrate goes into the formation of the enzyme substrate complex, or the breaking down of the enzyme substrate complex back to the enzyme plus substrate is called as K minus 1. So K1 and K2 are the rate constant. When the ES goes into the formation of enzyme plus product, it is called as the rate constant, which is K2. So if all the enzyme molecules are complex with the substrate, then the maximum velocity occurs and the Vmax becomes Kcat Et, where Kcat is the overall reaction rate and Et is the total enzymes to which the substrates have got bound. So this can be also written as Kcat is equal to Vmax by Et. So Kcat is one of the constant which gives us the idea what is the turnover number of an enzyme. So the turnover number of an enzyme is characterized by the Kcat, and with the help of the Kcat the scientists can decipher how many reactions are taking place at one active site within a per second or the unit time. So the Kcat is a very important constant for making out how exactly the enzymes are functioning. Now over here in this example I have shown the various Kcat values which are being shown over here. So if we see the carbonic anhydrase, which has got a turnover number or the Kcat which is per second, it is 6 lakhs, while in the case of the lysozymes it is 0.5, tryptophan synthase it is 2, DNA polymerase 1 it is 15, chymotrypsin this is 100. So this turnover number are showing the number of reactions which are taking place at one active site per second or per unit of time. Now the second important the constant which is associated with the enzyme kinetics is called as the enzyme efficiency. Now the enzyme efficiency is characterized by the term which is called as the Kcat divided by Km. So the Kcat by Km is nothing but K1 into K2 divided by K minus 1 plus K2, where K2 is small and the denominator becomes K minus 1 and Kcat by Km is small. So the enzyme efficiency is characterized by two major constants, which is Kcat divided by Km value. So if we can know the Kcat value and the Km, which is the Michaelis-Menten constant, we can figure out how exactly the enzymes are functioning or what is the efficiency of the enzyme. So the Kcat by Km, which is the enzyme efficiency value which is shown over here. So over here I have shown some of the amino acids in the ester and how exactly the enzyme efficiency is being shown with the help of the enzyme which is the chymotrypsin. So the chymotrypsin preference to the various substrate is shown over here with the help of the glycine, valine, phenylalanine. So it is shown over here. Now let's come into the most important part of the enzyme kinetics, which is the enzyme inhibition. So the enzyme inhibitions are broadly classified into non-covalent bonding, which is the reversible inhibition, and the second one is called as the covalent binding or the irreversible inhibition. The non-covalent binding or the reversible inhibitions is divided into three major inhibition processes. One is called as the competitive inhibition, the second one is called as the uncompetitive inhibition, and the third one is called as the non-competitive inhibition. While the covalent binding, which is the irreversible inhibition, is divided into three major classes. One is called as the group specific reagents, the second one is the reactive substrate analogs, and the third one is the suicide inhibitors. Now let's take up the first one, which is the reversible inhibition, which is the competitive inhibition. In this one, the substrate and the inhibitors compete with one another to bind with the active site. As the substrate and the inhibitor are having complementary or similar structure, they can bind with the active site. So what happens is, to the free enzyme, when the inhibitor binds, it goes into the formation which is called as the EI complex or the enzyme inhibitor complex. But when the substrate outreaches the inhibitor, it goes and binds to the enzyme active site and it forms the ES substrate or the transition state, which further goes into the enzyme product complexes. So let's see how exactly the enzyme kinetics are fluctuated, are regulated with the help of the competitive inhibition. So if we see the graphical representation over here, the Michaelis-Menten plot and the double reciprocal plot, it says that the Km value is raised and the Vmax remains the same in the case of the competitive inhibition. So we can see both the graphical representation over here. The one is showing the Michaelis-Menten plot, the other one on the right hand panel is showing the double reciprocal plot, while the double reciprocal plot equation is shown over here. Now let's come into some of the examples of the competitive inhibition. One of the most important example of this competitive inhibitor is the discovery of the methotrexate. So methotrexate was discovered by one of the Indian born scientists, Dr. Subbarao. So methotrexate is one of the most important drugs which is used for certain types of cancer and to treat certain rheumatoid arthritis. Now methotrexate is one of the competitive inhibitor for the enzyme which is dihydrofolate reductase. This enzyme DHFR is one of the most important enzyme which participates in the synthesis of tetrahydrofolate, and the folate is one of the essential part of the biosynthesis pathway for the purines and the pyrimidines. So these methotrexate competitively inhibits this particular reactions within the cancerous cells, or they inhibits the DHFR enzymes which are present in a higher quantity within this cancerous cells, and they block the cancerous cells, and the further biosynthesis of the purines and the pyrimidines cannot take place within these cancerous cells, and they are killed or they die off. Now, if we see the structure of the methotrexate along with the dihydrofolate, which we can see that there is a resemblance, and both these, the substrate which is the dihydrofolate and the methotrexate which is the inhibitor, have got a similar or the complementary structure. So they compete to one another to bind with the dihydrofolate reductase enzyme or the DHFR enzyme. So methotrexate injections are available in the market and they are given in certain types of cancer. Now we come into the second reversible inhibition, which is called as the uncompetitive inhibition, in which the inhibitor binds to the enzyme substrate complex. So the inhibitor doesn't bind or doesn't compete with the enzyme substrate binding to the active site, but these inhibitor can only bind to the enzyme when the substrate is attached to the active site, and they block the enzyme substrate complex. So the complex formed is called as the ESI complex, and this complex cannot go further for the reaction, and the reaction is stopped. Now if we see the enzyme kinetics over here, how exactly the enzyme kinetics changes over here, it is said that the Km and the Vmax value are lowered in the case of the uncompetitive inhibition. So if we see the graphical representation over here, the Michaelis-Menten plot and the double reciprocal plot, we can see the Km and the Vmax value have lowered down. In the case of the double reciprocal plot, we can see there is a parallel red color line which is shown, which is showing the uncompetitive inhibitor which binds to the enzyme substrate complex, forming the ESI complex or the enzyme substrate inhibitor complex, and the reaction is shown over here, which is called as the Michaelis-Menten reaction. Now the last of the reversible inhibition is called as the non-competitive inhibition, and in this non-competitive inhibition, the inhibitor can bind to the enzyme or the free enzyme also, forming a enzyme inhibitor complex, or the inhibitor can bind to a enzyme substrate complex to form the ESI complex also. So over here, if we see the reaction, the enzyme inhibitor can form a EI complex which can further goes into an attachment process where the substrate is bound to form the ESI complex, or the inhibitor can bind to the substrate bound complex also, which is the ES complex, and it forms a ESI. So it can bind both to the free enzyme as well as to the enzyme substrate complex, and we can show over here, we can see over here the non-competitive inhibitor is bound to other site other than the active site, where the active site to the active site the substrate is bound. So the inhibitor binds to both the enzyme, free enzyme, or the enzyme substrate complex also. Now if we see the reaction over here in the enzyme kinetic, it is being seen in the case of the non-competitive inhibition, the Km value and the Vm, the Km value, which is the Michaelis-Menten constant, remains the same, while in the case of the Vmax, which is the maximum velocity, it is lowered. So we can see over here in the Michaelis-Menten plot, we can see the green line show the representation of non-competitive inhibitors, in which the Km value remains the same while the Vmax lowers down, while in the case of the double reciprocal plot, we can see the non-competitive inhibitor, which is shown in a red colored line. So the Km value remains the same while the Vmax value is lowered. So the equation is shown over here, which is the double reciprocal equation. Now there are certain irreversible inhibitions which have been also found in the case of the various reaction processes. These irreversible reactions are basically the glue-specific reagents which reacts with the specific R groups of the amino acids. So over here, if I show the example, the DIPF, which is the diisopropyl phosphofluoridate, it inactivates the acetylcholine esterase enzyme that terminates the synaptic transmission. So DIPF belongs to the group of compounds that are known as the nerve agent. So over here we can see the acetylcholine esterase enzyme, it is being blocked by a DIPF, which forms a covalent bonding over here, and this enzyme is permanently inactivated. So the inactivated enzyme we can see over here is bound with the DIPF and it doesn't function properly. Now there are certain reactive substrate analogues also that functions in the case of the irreversible inhibition, which are shown over here. So the affinity labels are the molecules which are coming under the irreversible inhibition. So the affinity molecules labels are the molecules that are structurally similar to the substrate for the enzymes that covalently modify the active site residues. So over here we can see there are certain affinity labels or the natural substrate for the chymotrypsin, which comes and binds to those particular enzymes and they irreversibly inhibits those particular enzymes over there. Now another of the example which is shown over here, which is the TPCK, which is the thiosyl phenylalanine chloromethyl ketone, which is shown over here. So this is one of the analog for chymotrypsin substrate which binds and irreversibly inhibits the enzymes over here. So if you see the diagram over here, the chymotrypsin is attached with the TPCK, and this TPCK which covalently binds with this chymotrypsin and it inhibits or the irreversible inhibits the enzyme over here. So these are some of the examples of the irreversible inhibition, and the last example which comes up is called as the suicide inhibitors. These inhibitor binds to the enzymes as a substrate and initially they are processed by the normal catalytic reactions. The chemical reactive intermediate inactivates the enzymes through the covalent modification. So what happens is initially the inhibitor binds to these enzymes and the reactions takes place. So within the reactions, the normal catalytic mechanism reactions takes place, and subsequently the product which is formed over here, this product becomes suicidal inhibitor for that particular thing and it reacts. So the example over here is the aspirin, which acts as the covalently modifying enzyme for the cyclooxygenase. So these are the various inhibitions and the enzyme kinetics which we discussed over here, and with this note I come to the end of my lecture over here. Thank you very much.
Thank you, sir. Thank you very much for this productive session. Thank you, viewers, for watching our show. We will be having more lectures in the current lecture series. Till then, goodbye. Take care. Thank you.